Hi everyone, my name is Salvador Casani. I'm a last year PhD student and I'm going to show my work on a multi-omics approach to characterize the yeast metabolic cycle using multivariate statistics for omics integration. My poster number is 149 and I hope you like it. So the yeast metabolic cycle is a model system in which around 60% of the yeast transcripts cycle. These transcripts oscillations have been characterized in three well-defined phases that have functional implications for the progression of the cycle. The regulatory mechanisms by which yeast cells manage to control cycling of transcripts is now under research. And one of the main contributors of these changes is the epigenetic landscape of the cell. Metabolites in this particular system also show an oscillatory behavior. And since metabolites have been appointed to contribute to the epigenetic modifications, we wonder if it was possible to observe a cross-regulation between the three metabolic molecular layers in a way that metabolites may be changing epigenetic landscape, and then epigenetic landscape changes gene expression of the yeast metabolic cycle. In order to do this, we extracted a multi-omics dataset in which these three layers of molecular information were present. All omics were processed in a similar way and subjected to differential expression of features, and clustered in three different clusters that has been described in previous yeast metabolic cycle studies. Although all omics could be modeled in these three clusters, histone modifications ATAC-seq and NET-seq show a much less support for RB phase, making us doubt that the epigenetic regulation of the cycle is happening in this particular layer. Once the omics were processed, we proceeded to their integration to try to unravel the regulatory mechanisms that link them. The use of multivariate statistics is especially interesting to model biological rhythms. In these rhythms, there are many genes that coordinate their expression, and with multivariate statistics, we can model the general trends of each omic as a function of the other. We used PLS to model metabolites' influence in gene expression. We could plot the overlap of their loadings, which allows to see which metabolites are accumulating at each position of the yeast metabolic cycle. For example, we see that at the end of OX phase, which is the red phase, there is an accumulation of amino acids. And at the beginning of RC phase, which is the blue phase, there is accumulation of NAD-related metabolites, which are blocking sirtuin activity, for example. So this could allow us to make functional interpretations of the biological mechanisms that are going on here. We used MPLS to model histone modifications' influence on gene expression. This allowed to select the two histone modifications that have the highest influence in the regulation of gene expression or the modeling of gene expression, which are H3K9 acetylation and H3K18 acetylation. And finally, we used ATAC-seq to model gene expression, which shows that we model gene expression in two ways. One using peak quantitative models, which is the figure on the right. And with the peaks, we used PLS to model gene expression. And we see that the gene expression is separating in two main phases. Whereas when using the transcription factors that bind to each peak, we build a generalized gene regulatory network, and we see as well that the two phases in which the peaks are acting mostly or transcription factors are the red and the blue phase, which correspond to OX and RC, and are the phases that correlated best as well with or were best shaped in the other omics, at least in the single analysis, which make us doubt of the regulatory mechanisms that are behind the RB phase or the green phase. The pairwise modeling of the omics was nice, but we wanted to combine all the omics together in one model. Our first approach to combine omics was done by creating generalized linear models between the metabolites and the other omics, so RNA-seq, NET-seq, K9 acetylation, and K18 acetylation. This showed a regulatory network in which RC phase was the predominant phase of the cycle and gives importance to aspartate and asparagine as the metabolites with most connections with other omics. But what we really wanted to do was to model the flux of information that is coming from metabolites to chromatin activity and then how this is translated to gene regulation. And this flux of information was modeled in our main contribution to the field, which will be the application of PLS path modeling. This is a structural equation modeling tool where we can define latent variables for each of the omics. Each latent variable is defined by their indicators, and the whole model is then subjected to a series of iterative calculation of coefficients that selects the best indicators for each latent variable and deletes the relationships that are not supported by our data. This allows the selection of metabolites that are more relevant to explain variability in the subsequent regulatory phases. One of the latent variables had to be split in two, as you can see here. They're in early RB metabolites and late RB metabolites because they have a very different patterns, as it can be appreciated. We also randomized relationships in 1,000 models and plotted the goodness of fit, showing that our model is highly relevant compared to the distribution that we obtain. A big advantage of using the PLS path modeling is that we can see which are the best indicators on each of the latent variables. In this way, we can select the features that contribute the most to our model, and these features are putatively one of the drivers of each phase or each of the positions that we model of the yeast metabolic cycle. Finally, these are our conclusions, and the most important part or take-home message from here is that with the PLS path modeling, we can define all the molecular layers that we want to define in our model, and we can see which of them contributes to the cycle progressions through molecular cross-regulation. We can thus identify metabolites that could influence the yeast metabolic cycle. And that's all. Thank you very much for your attention. And if you have any questions, there is a lot of condensed information here due to time restrictions, but you can find my email in the poster and also my Twitter account. Thank you very much.